Hey guys, sorry about the casual apparel, but the idea to do this video was kind of a last minute decision. I was going to do everything just kind of with written instructions, but I thought you guys might benefit from my usual little pre-lab spiel in our lab on urinalysis. So a couple of things. Urinalysis is one of the oldest diagnostic tools in medicine. This goes back many, many centuries before we had, fancy little instruments like this and chemical dipstick tests like this. Physicians would use their senses to evaluate a patient's urine and make determinations about their health. And one of you are going to be doing this to some extent, albeit the only sense you're going to be able to use, because we're doing it virtually, is your eyes. But just like you would assess a diamond based on clarity and color, you can do the same thing with urine as well. Urine should be pale yellow, pale to straw yellow, and it should be clear. It shouldn't be cloudy or turbid at all, okay? So when you guys are analyzing our virtual patients, which is not really going to be all that different than what you would do in lab, you're going to look at a sample of the urine in a clear tube on a white background, and you're going to be able to make some assessments on the color and clarity of that urine. Is it clear, cloudy, turbid? Is it pale yellow, straw yellow, dark yellow, amber, orange, red, black? Believe it or not, urine can come in all of those colors. Now, the other thing old physicians would do back in the day is they would smell the urine. Oh, I spilled a little on my desk. They would smell it. Fresh urine should have very little smell unless somebody is ill and there are accumulations of things like ketones or aldehydes. Sometimes people with diabetes, you'll have aldehydes build up in their bloodstream. And even back in the day, the physicians would give it a little taste. It's in real urine, y'all, by the way, just so you know. This is not real pee-pee. It's just water I put some yellow food coloring in. But the main thing they would taste it for is sweetness, to see if a person were diabetic. Now, your kidneys should, through tubular reabsorption, get back all of the sugar that gets filtered out in the glomerulus. But people who are diabetic, whose blood sugar is artificially elevated for a really long time, have so much sugar in their blood that even clearing out as much as they possibly can, they can't get it all out. And so some of it ends up in the urine. So the first thing you guys are going to do is a visual inspection of the urine. For each patient, 1, 2, 3, or 4, on your little handout, you're going to be using your lab manual like you normally would. What I want you to do is fill in the color and the clarity. Is it clear, cloudy, turbid, whatever, based on what you see in the pictures. Now, the next test that I pre-performed for you and just took pictures of it was specific gravity. Specific gravity is the measure of a density of a liquid compared to the density of water. So water is always going to have a specific gravity of 1.0000. Okay? Urine, because it has stuff dissolved in it, is going to be slightly more dense than water and will therefore have a slightly higher specific gravity, but it's going to be really, really, really low. Your specific gravity values are always going to start 1.0. And really, you're going to use this device, a urinometer, to fill in those last two numbers. So you might want to go ahead, pause the video, and on your handout, go ahead and write 1.0 in every single space for specific gravity. You're going to measure it twice, once with the manual version and once with the chemical dipstick. Okay? But what you would do is, if you guys were doing this in lab, I got to hurry because I want to try to keep this video as short as possible, is you would pour some of the urine into the tube with the urinometer probe until it just starts to float off the bottom. You can see now it's floating, it's not touching the bottom. And then you would look at where the meniscus, the air-water interface, touches this tube. Now, because this is just water out of the water fountain, you can see this one is not floating very deeply at all. It's hitting right at 1.0000. I know that's a little fuzzy, but that's the best my silly webcam can do there. You can see it. And it's going to float. The more dense this is, oh, I'm dripping on my desk again. The more dense this is, the higher up this thing's going to float, and you're going to read the number accordingly. Now, there's a section in your lab handout. I'm sorry, it's behind me like you can see it. In your lab handout, there's a little section that tells you how to read these. And in fact, one of your homework assignments this week is I've got a lot of practice opportunities for you to read kind of a virtual urinometer probe. But you're going to record the reading that you get from the picture that looks like this thing under specific gravity 1 in your handout. And then the next thing you would do if you were doing this in class, pour this all back out, is you would use one of these. So this is called a Multistick, and it's got 10 different little absorbent pads on it. So I'll pull one of these out so you can see what it looks like. And what you would do if you were in class is you would take your urine sample, you would take this, you would dip it in, and then after a predetermined amount of time, you would compare the color on these pads to the color on the label of the bottle. So what I would do, for example, if I wanted to read glucose, is after 30 seconds, you can see it says 30 seconds right there, I would compare this bottom tab, and we can see it's closest in color to this first box there, this one right there, so it's negative. This one doesn't have any glucose in it, which is good because it's water from the water fountain. So what you're going to do then is you have in your handout one of these labels laid out nice and flat for you, and you're going to compare the colors in the picture of the dipstick multi-test to the colors on your label, and you're going to fill those into the appropriate cell for each patient in each test in your handout. Finally, the last test that we could do here is microscopic analysis. There are suspended sediments in your urine, shed cells, mucus threads, microbes. Urine shouldn't be considered sterile because you can have microbes in the distal portions of the urinary tract. So for each patient, there's going to be a characteristic micrograph, and there's a link down below of a bunch of different things that you might find in urine sediment. So compare what the patient has in their picture to the illustration in the link below and see if you can figure out what they have. Finally, you're going to use all the information in the patient presentation, like what was their major complaint, why did they come to the doctor, all the information in their urinalysis, and you're going to diagnose them. Now keep in mind why they're there. If somebody might not have a disease or a condition, their urine might be clear, they might be perfectly healthy. So keep that in mind as you're evaluating these patients. When you're all done, I'm going to have a little thing that you can do so that you can see the answers and make sure that you are good to go on this. And as with anything else, as we've moved to online, I am available via Zoom conference pretty much unless I'm in class with you guys on Tuesdays and Thursdays, or with my parasitology class on Mondays and Wednesdays. I'm available. I might not be right at my computer, but you can email me. You can DM me on Facebook, Instagram, or Twitter. I'm also going to try to link my office phone to my cell phone. So if that works, you can call my office number. We'll find out if that works early next week. But don't hesitate to reach out, and I'm more than happy to tutor you one-on-one or in small groups pretty much any time as we move forward here. I really want to stay active and as involved with you guys as we can. All right? Well, that's it for now. Have fun, stay safe, make good decisions, don't drink and drive, and remember, only yes means yes.